Hello, Science 10, and welcome to our lesson on cell membranes. This is lesson 3, and today's goals are all about learning about the outer layer of our cells called the cell membrane. The cell membrane is really, really important, and we're going to talk about it for pretty much the rest of this chapter. So today you're going to learn about the function and importance of the cell wall and how it helps maintain life, and we call that homeostasis or balance. You're going to be able to explain why smaller size cells are better than bigger size cells. I need you to be able to define the words phospholipid, hydrophobic, and hydrophilic. You're also going to have to tell me what is a phospholipid bilayer. And I also want you to tell me how cells maintain homeostasis or balance of our bodies by regulating movements of ions, proteins, and nutrients in and out of the cell. So cell size is probably the most important concept right now, which is essentially you now know what a cell is. We know how to see a cell with microscopes. We know we can stain cells such as bacteria. But why are cells the size they are? When we talk about cells, we know they're super small. Why are they super small? Well, cells are very, very small because they need to move waste out of them, but also bring energy into them. So their size matters. All right. Think of an Olympic-sized swimming pool to like an inflatable pool in your backyard. If you had to swim back and forth across an Olympic-sized swimming pool, okay, to drop things off on one side and then pick something up and swim it across to the other side, you're going to tire out really quick. Compared to your little inflatable pool, where if you climb into your inflatable pool and you sit down, you can almost just reach out and put things on sides. Okay, it's way easier. Cells need that because they're small and they expend a lot of energy to do small things. So we say substances diffuse. Now, diffuse just means moving along a concentration gradient. We're going to talk about that in a couple of lessons. But they slowly diffuse these things inside because they have very small concentration gradients because they're really small. Now, that part will make more sense when we talk about diffusion. But what you need to understand today is that cells, the most important thing for them is to have a large surface area to volume ratio. So what do I mean by surface area to volume ratio? Surface area to volume should be as big as possible. All right. So if we look at this small cube, imagine it's a small cell. Our surface area, each of those sides are about 1 millimeter. Okay. And if we multiply the 6 sides together, we get 6 millimeters squared. Essentially, the surface area. If we look at the volume, okay, that's cubed, 1 times 1 times 1, because they're all 1 millimeter, okay? That gives us 1 millimeter cubed. So our surface area to volume ratio is 6/1, okay? So this ratio means that our cube has a 6:1 surface area to volume ratio. So its surface area is much bigger than the volume inside the cell. Let's look at this large cube now. Imagine it's a large cell. I said cell, not really meaning to. But let's pretend that the surface area still has 6 sides, but now they're 2 millimeters instead of 1. Now all of a sudden we have 24 millimeters squared of our surface area. Perfect, it's quite big. That's a big surface area. But if we think about the volume, we have to multiply each side by 2, because it's 2 times 2 times 2 will give us the cubed. We'll get 8 millimeters cubed. So if we look at the formula surface area to volume, it's a 24:8. Now that looks big and you're like, oh, that's a really big surface area to volume ratio. Except we have to put in lowest terms because both of those are divisible by 8. And you actually only get a 3:1 ratio, which is much smaller than our 6:1. So the surface area is much smaller compared to the volume inside of it, which again is a problem. We want the largest amount of surface area, the largest amount of space for things to come in, but we want the smallest area inside for things to move around it. So what happens when you have a larger surface area? Okay. Well, if you don't have a large enough surface area to volume ratio, you will die. So the larger cube will not survive. Okay. Like I said earlier, this will not be able to move nutrients in and out, and it's not going to function efficiently. It'll use more energy than it creates. All right. So that's a really big problem because a lot of cells are kind of a little bit bigger or belong to structures that are bigger. So some organisms actually have physical structures attached to certain areas to help increase their surface area to volume ratios. Plants, which we're going to learn about later, have these things called root hairs. They're really, really skinny and tiny, and we've seen pictures of them. They're about 15 nanometers I think, in size. And essentially, they create more space on the root so that you can absorb nutrients and water. Your intestines, your small intestines, which is what you see there is a picture, is actually made of tons and tons of folds. So your small intestines is a bunch of little folds. And within those folds are these little finger-like projections that have little tiny projections off of them even. And all of that creates more surface area, more area for nutrients to pass across and be absorbed in. So again, these increase our surface area, which is important because our volume is so large. So we want to increase the surface area to ensure that we are efficiently absorbing nutrients and getting rid of waste. So now I keep saying that this is really important because things are going to pass into the cell. Well, where are they passing into? And what are they doing? Well, cells are what we call open systems. Okay, open systems means that matter has to get in and out of the cell. If it was a closed system, nothing would leave. But our cells are open. They want energy in and they want waste out. So we have something called a cell membrane. A cell membrane is designed to separate the cell from the surroundings around it. So whether it's a liquid outside or it's a cell in your intestine, it's just a membrane that keeps all the organelles inside and protects it. Now, for your membranes, cell membranes, we're going to specifically talk about cell membranes being made out of phospholipids. Phospholipids are the main ingredient of the membrane molecules, and they're embedded and attached. And there's other molecules that are embedded and attached in between these, and we're going to look at that here. Every cell membrane is what we would call semi-permeable, okay? Now, semi means a little bit. Permeable means allowing things in. So it somewhat lets things in. This means that our cell membranes only allow certain things into the cell while blocking other things from coming in. So if you look at this picture, I have red blood cells, I have large proteins, and I have glucose and maybe a little bit of urea and some chlorine. You'll notice the bigger the structure, the less likely it is to pass through my cell membrane, which is that orange structure right there. What's happening is that cell membrane is blocking blood from entering to inside the cell. It's blocking proteins from entering inside the cell. Why? Because blood and proteins don't need to be in the cell. The cell doesn't use it, so we don't pass it through. So that's what it means by semi-permeable. It means only certain things can get through into the cell. So let's talk about what is making our cell membrane up. This is called a phospholipid, okay? It has a hydrophilic head and a hydrophobic tail. What the heck does that mean? Hydro means water. Phobic means fear. Philic means love. So when we say we have a hydrophobic tail, we have a water-scared tail. So the two structures that you see there are actually made out of fats, and so they are not love and water. Think of mixing oil and water together. They don't mix, okay? But our hydrophilic tail, which is made out of something else, hydrophilic head, which is made out of something else, likes to be near water. It dissolves readily in water. So when you place the hydrophilic tail in water, it dissolves readily. Hydrophobic does not. Why? Because it's scared of water. The chemical interactions causes the phospholipid bilayer to form. So what you will see here, and I wonder if I can get this to work, is you're looking inside at the plasma membrane. This is a cell, and the structure around it is the cell membrane, which is super cool. So we're going to learn about all those things inside, but let's take a closer look at the cell membrane itself. You can see there the hydrophilic head and the hydrophobic tail, okay? And they sit tails pointing in away from the water that is inside the cell and outside the cell, so they face inwards. And then the heads face outwards so that they can be in contact with the water. So it creates some sort of layer like this. So kind of think of a bread on a sandwich, and then the things in between are the tails, which is the inside of the sandwich. So this interaction forms what we call a bilayer. So cell membranes have also things embedded in them called protein channels. And protein channels, ladies and gentlemen, are super, super, super cool. And I cannot wait till you come to Bio 20 and 30 and we get to talk about some really cool channels. But essentially, they're found stuck in between all the phospholipids, okay? And their job is essentially to be there to support bigger structures to coming into the cell. Why? Because sometimes we do need bigger things inside, okay? Sometimes, like sodium and potassium, are too big of ions to make it inside the cell. But if you don't have sodium, potassium rushing inside of a cell, You're not going to have nerve impulses occur. So you would never be able to lift your hand or kick your foot or blink your eyes. So sodium potassium channels are essentially protein channels that allow sodium and potassium in and out of the cell because they're too big to pass through the phospholipid bilayer. So these proteins, like I said, have to move substances across membranes. They also can help carry out chemical reactions. Like I said, big one is neural impulses. Photosynthesis uses channels, protein channels, to bring hydrogen in so we can create energy for photosynthesis. Very important. Okay. They actually are used to identify other cells, which is kind of interesting, as well as protect cells from infection, and they receive messenger molecules to help pass on information. Okay, so in the example I kind of just talked about was the sodium potassium channels. But again, guys, it's just about moving larger substances across the membrane because they cannot get in to the membrane without help. So the membrane function prevents, like I said, many substances from entering. Ions, salts, sugars, proteins, viruses, and bacteria. You know, one's living and one is not. Okay, that's its role. All right, it's trying to protect the entire cell from having anything enter into it. Okay. Also, not only do you have membranes on the outside of a cell, you have membranes on almost every single one of the organelles inside your cell. So that means that your mitochondria, your chloroplast, your nucleus all have membranes that also surround and protect. Okay. Those membranes help promote necessary reactions, and they also protect and prevent unwanted reactions or things from getting into them. So another important part to remember is cell membranes are all about regulating movement of substances in and out of a cell. Okay. Cells membranes have to be semi-permeable. Otherwise, it's not going to work. Everything's just going to be allowed to be in the cell, and we don't want that. Okay. We want small neutral molecules to be able to cross the membrane, but we want to stop other things that we don't want, or we want to be able to give them help, like protein channels. So this video is a little bit longer.
All animal cells are enclosed in a plasma membrane, which consists of 2 layers of phospholipids. The hydrophobic nature of the cell membrane makes it intrinsically permeable to small nonpolar and uncharged polar molecules, but non-permeable to large polar molecules and charged particles. Charged particles, such as ions, must use special channels to move through the membrane.
Okay. So that's kind of what's happening, okay, when it comes to the cell membrane. So hopefully you have a little bit of an idea about cell membranes, because when we get to talking about transports, you're going to need to remember that there are channels, that the cell only allows certain things in. And when it does, there's a couple different ways that can happen. So thank you, and I hope you have a great day.